Okay, good morning. I hope you all have a good spring break. Good. So this is a week number 9. We are going to discuss regression, and also we will learn what is a regularization. But instead of going through the slides, I'm going to do a way upside down. So we look at the functions, look at the coding, and see how we can do the regression, simple linear regression, multiple regression, and even generalized regression. And then use small examples to play with those functions in MATLAB. Then we will look at those applications in single cell data analysis. I can think of at least 3 different application areas in single cell data analysis. So once we have problem and say, okay, there are so many variables, parameters in order to predict the outcome. So how can we do this feature selection to find out most informative variables to explain the outcome variables? So that's the problem of lasso and the regularization. You always have problems to pronounce this. So I'm not going to go through slides to introduce theory. Instead, I'm going to leave those theory part dissolved in the coming weeks, Tuesday lectures. And then the second hour, we will discuss this application for it was a research, and it's about highly variable gene and see whether they're functionally important, or whether they are just a gene expressed highly noisily, or there are some implications in terms of the function of cells. Okay. So as a starting point, we still have to update our package, because during the weekend, I implement several functions, including those functions to do the meta visualization. I shared the paper with you. It's about you have a TSD, UMAP, fate, whatever embedding. They are not consistent. How can you synthesize, put them together to generate this so-called meta visualization method to learn from all the different kind of embedding to give you the better resolution, probably. I haven't tested this in real applications, but the function has already been implemented and provided in toolbox. So if you have the problem, say, I use UMAP, get this, and TSD get that, which one is the best? So now you can put them together. Okay, use the resolution provided by multiple different embedding algorithms. And we can see how good the outcome is using when you use real data analysis in your data. And we can decide whether I will put this as a by default option in the package, right? I still have to wait until we have some practical experience with this method. Okay, so regression, I don't know how much you had experience with, but this is a let's see, it's supervised or unsupervised. Do you think it's a supervised algorithm? It is. I think the purpose of this regression is you collect your sample, you have data points, and to learn the relationship between variables. And then you want to use this as a foundation to predict future income data points. In that sense, it's supervised learning, but the process itself, you just have to collect the data and then learn the relationship between two variables. The process, I don't think is supervised. Anyway, depends on the use. Okay, for example, my example is if you have two genes expression across cells, and if you want to know the correlation or regression relationship between these two genes, so if you can use one gene's expression to predict another gene's expression, then you do this regression, then you establish relationship, you learn the regression coefficient. So I don't think this is a supervised learning. Let's see. Okay. Yeah. We no, if you do the linear regression, you have a linear model. You can have an interaction term. I show you how to do it later. It's not a model. It's a linear model. Yes. Yeah. Okay. So as I said, we jump directly to how to do. Let's do how to do it. Okay. So let's do a documentation. So that's the function to do the linear regression. Let's read the documentation. So what's the input, what's the output. By the way, you have a homework due today. I know some of you complained about you don't have enough time or something like that. You don't know what to do. That's fine. Yeah, so I want you to do to build a classification model, and the input is very similar to this. You have an X, which is a matrix. It's in your spreadsheet. And then Y is indicators, say yes or no, so the binary, so whether this sample depends on its variable, it's a patient or healthy individual. So the setup is very similar to this. Instead, you replace regression into some classification. Yeah. Yeah, so if you need more time, I will see how many submit, and I think it's good if you found Yunjian to talk about what to do. Do the suggestion, or you don't know what to do at all. Yeah, but still. Do we have a classification learner? I remember. Is it useful? Yeah. So if you have found a way to get this big X matrix in there and a Y variable, the dependent variable, maybe just try this. Maybe I don't know. I haven't tried this. Maybe big data set is too big for classification learner. You can use this to do it. Same thing here is we also have a learner, regression learner. You see? Okay, so we will learn how to do this using command line to do this regress, and then we can use regression learner to see I import data and to learn. But here is just tell you how to do it, but I haven't explained this inside this regression function. What's inside there? How the computer algorithm is doing in there. So that's involved in the multiplication or optimization. Okay, so you search for the parameter combinations in order to get the best solution. Okay, no theory so far, and just look at the description. So, so this function will take big X and Y as a respondents. Okay, it's the value you want to predict using X. And the B return coefficient estimation. So B is a beta. So if we are talking about a linear regression, so you regress and obtain a line. This line will be defined by its beta value. Okay, the slope. So beta basically is a slope with intercept. Okay, we don't need to know this confidence or the other residuals. So those are the other products you can produce from this function. So normally in MATLAB, if you have a capital variable name, means it's a matrix. If it's a small case, it means a vector. So in this case, we have a matrix to predict a vector. Okay, let's look at an example they provide. They provide this data from the car small, so we can load them and then assign the weight and the horsepower into two variables named x1 and x2. What we want to predict by giving x1 and x2, so x1, x2, you can put them together, put in the big X, okay? We want to use these two variables to predict miles per gallon, okay? So that's your outcome Y. So what you can do is you copy this and put in here. Then we have some variables to play with. Okay, so this is our Y from a sample of 100 different cars. 13, so there's some really highly efficient cars, and you can miles per gallon, so you can run 38 miles per gallon. That's really good. And we can see there are some missing values. Okay, the function will take care of these missing values. It will be get rid of. Okay, so some of the sample is invalid, no data. We will not use them. Okay, so now let's look at x1. X1 is this. So x1 is the weight. We can first see we can do a scatter plot. Okay, then we put x1 here in the x-axis. Just have a quick impression of their relationship. Okay, everybody can get this very easily. So this is using one variable, x1, to explain y. So normally we put a response variable into the y-axis, and x is what your independent variable. So you want to use variable in x-axis to explain y-axis, okay? So now we can see they are negatively correlated. If you have a car very heavy, then the miles per gallon level is low. That's expected. So we do have this very clear causal relationship, okay, with the C. So now our goal is if you want to use only one variable to explain another, we only need one parameter. Basically, we want to know the slope if you have a line here. So for example, we can see a basic fitting in the 2 here. Yeah, so if you want to use linear fitting, okay, so this is very simple linear fitting. Give you a line, and this line goes through all the data points. So how this line is defined? It's defined by 2 parameters. One is this. You see this? And then another is intercept. So it have to be the value to tell you where to put what if you have access at a 0 point here. Okay. So how these 2 numbers were computed? It's by optimization. Okay. They try to optimize. So if you change this value, negative or positive, or give different, you change the direction of this line here because the slope is different. So you want to achieve all the predicted value from the observed point to this line to be minimized. Okay. Okay. So you found the difference from this by giving the fixed x value and what is the y value as a prediction on this yellow line. So you want to make the overall difference of those observed points y to this predicted value in total as small as possible. All right, so that's linear regression. Yeah, so if you want to plot out the difference, so this error basically is the residuals. That's the difference between, for example, this point, the difference from observed blue dot, blue circle to this prediction is very small, it's very similar, right? The difference will be here and close to 0. Okay, that's a residual. Let's find another point like this. Okay, you observed to prediction is very different. It's negative values. It's corresponding to this point. Okay, so this plot tells you if it's all close to 0 here in the middle, means your prediction is really good. Otherwise, if you have a dots, which is a residual plot, the dot jumps everywhere. So the overall difference from your prediction to the observation is very different. So you have the sum of total of this difference. Okay. So this solution gave you the minimized fitting to minimize all these errors. Is that clear? Okay. Now next, So next, in this example, they create this big X matrix. They put X1 here, X2 next to each other, and also the product of X1 times X2. So in this way, there are three variables introduced. So they want to use three variables to explain Y, okay? So it looks like there are some of the variables which you cannot explain very well with one, the weight of the car, but another, when you combine them, give you better resonance, better fitting. And this term tells you if there's interaction between these two variables. To create a new variable and multiplicate them. And they can learn whether this will also contribute a lot to your regression model to explain why better. Okay, this is a term which contains all ones. It's the same size of this individual variables. If this have 100 variable, 100 sample, this will be 100 ones. So the purpose for this is to allow regression function to learn the intercept. Okay, this will give you a value. This is the one we just see 49 something in the linear regression of the plot. Okay, so in that application, the plot I click, it automatically adds this term in their internal algorithm. So we need this to get the intercept. Otherwise, you only have the rotation to have the slope. Then you don't have this offset. So unless you normalize everything here into have a mean to the zero, otherwise you need this term to give you the better resolution, not the better resolution, the fitting, overall fitting. Okay. So we can try to construct X. So you can construct with the interaction term or not. So I'm going to construct this without interaction term. Okay, that's my X, big X. So I have this column all ones, and then I have X1, that's which is the weight, and X2 is horsepower. All right, so I will do to give me B from regress my X. Oh, put the Y first. Okay, so I finished. This is called a multiple linear regression. Why is it called a multiple? Because I have two variables used to explain B. If I only use one, it's called simple or single variable regression. The multiple linear regression, and here I get three variables, the beta value, and here the example has 4. Why? Do you know why? Why they have 4 values in the beta outcome? The B, I only have 3. Yes. So the number of this B is corresponding to each of the columns here. So I only use X1, X2, and the intercept term to get 3 values. This is the intercept for this this is beta value for this variable, this beta value for that variable. Well, in this example, they use 1, 2, 3, 4, 4 terms, including this interaction term to get 4 values. And at the end, you see this interaction term have beta 0. So this one plays no roles at all to adding any values to help you to explain the variable. So eventually you can get rid of this, okay? Because the beta value is too small. Contribute nothing to help you to build a better model to explain why. So you can copy this and this piece of code. Yep. I cannot do it because they have a beta 4 value here. But anyway, if you follow the example before, you included that term. Let's include that term. Okay, then I can plot this. So it depends on these two variables combinations, and you define your data points here, and then you can predict what's the miller of per gallon by look at the combinations of these two here. So that's why you have this plan here. Okay, so that's your observed data points around this prediction plan here. Any questions so far? The rest are not so important because there are some this look at the place with residuals. Now let's just use our x and y. So we have x and y now. Let's use regress linear. Regression. I clean my workspace, close. So this interface provides you access to not only the linear regression, but many other regression methods. So we are going to just use linear regression for illustration purpose. Now we need to have a new session and we need variables from a workspace. Okay. Then you have to be very clear and which is your input variable and what's your response. So we want to use dataset X to predict the variable in workspace, which is Y. Okay, you select those. but give you 1 minute to finish this step. So the X is already in the workspace. We created the X. The contains 4 columns, right? So that's the Y. You can, let's see if I can rename this. No, I cannot. Okay, everybody reached to this step already. Okay, we can. Oh, the cross validation here. You don't know what that means here. Yeah, so this is want to give you some idea whether the prediction is robust, accurate enough. So what this will do is to maybe cut the data into half, use the top 50 samples to predict, and then use another half data to validate. So, okay, the slope you found, it works fine with the top 50 random samples, but not the rest. Something like that. Okay, in the center of the plot, you see those are the record number. That's just the sample you put in. We have 100 samples with some missing values. And then this is their Y, so there's a miles per gallon. Then you can look at other variables, but you can change this x-axis value to column 2. Remember, column 2 are the weight. So you get a scatter plot between column 2 variables with response. So this gave you a way to look at your data. The horsepower, the production, the multiplication between the two, so nothing adding on to. Okay. Then here you select. There are many other algorithms to do the regression. Okay. What we can do is to select linear, and then is to do the training or run the model. Yeah, so that's a the solution, basically, this problem. The, uh, residual plot looks like this. Okay. A lot of things we can play with, and there are specific models that allow you to do this interaction. If you have multiple, say in this case, we only have x1, x2, so very easy to do this interaction. So x1 multiplied by x2. So if you have x1, x2, x3, then there's more than one interaction term. So you may have to do multiplication between x1, x2, or x1 or x3, or x2, x3, or maybe all the production. So I think this model will help you to do this combination. Okay. So now is the problem. We come back to single cell data. So I'm going to close everything here and then see how can we close F. I just clear everything and then G2. So I'm going to get this single cell data. I just use example data and then export variables to workspace. I will select this two. It's the expression matrix and the gene name. Okay. Now we have more than 7,000 genes. So we have in the big X, more than 7,000 rows. What we can do with regression? Any questions? Okay. No. Got this. Is we can do this pairwise regression. So I can, for instance, the Y, which I want to predict, is one of the genes expression, say 134 number gene. Okay, you can extract this gene expression, and then transpose it into a column vector. Yeah, so there are a lot of zeros anyway. Now, if I have X1, another gene, say, random number, okay? Now you know how to do the regression between these two. You want to use X1 to predict expression of Y. So what is X1? X1 is gene number 4, 5, 6. So let's look at what's the name of the gene. So you want to use gene ARL5B to predict expression of gene 134. Is that clear? Making the case clear. Is it a clear case? What do you want to do? You want to use one gene's expression to predict another gene expression. By chance, I just select the random 2 genes, but you can select any pairs of genes to do this, right? So what's your command line here? You can do 2 versions, one with intercept, one without. So what you can get from the information from the B data? Do you have this? So which value is important for us? Is this intercept? No, is this a slope? I don't think this scatter plot will look pretty because there are so many zeros in there, but at least this should tell us some of this relationship between them. I care about whether this is positive or negative. Okay, positive means the line is going up. Otherwise, it's negative. It's like the horsepower and the minus, and then it's negative. I don't think this scatter plot will look pretty because zeros there. They all a lot of zeros. Okay, when you use this, I think one thing you need to remember is all the column vectors. Okay, so you cannot have I don't know if you can put a vector which is 1 to n, and it has to be n by 1. I think so. And then you have each variables in the column. So that's why we have to transpose the original rows from the big X matrix, because each row is one gene. We have to transpose it in there. So let's see whether we can improve this by adding other genes. So maybe I can use top 50 genes. Remember to transpose this. So it was just one gene, X1, X plus Y. Now I'm using 50 genes from the matrix. This extract 50 genes and then transpose it, put the genes in the column. So I get my beta becomes 51 values, and then the first intercept, and then each corresponding to the 50 genes explanation power to predict the Y expression. So you use a lot of genes to predict another gene's expression. And this is which one the beta value, absolute value is bigger, means the contribution is more stronger. Okay. Now the problem becomes, can we use 5,000 or use all other genes per gene? Well, I shouldn't include Y itself, because the Y is from big X itself. So I should exclude Y. Y is which genes? 130, I select 134. I can't remember which gene I get for the Y. But if you use 5,000 genes to predict if that gene is in there, this particular Y gene inside here, that gene will give you a perfect prediction, right? We can try and see which beta. So I forgot which Y I 134. Then if I do the top 500, then see beta value, which one is the largest. I will do absolutely beta value and the max is 1, and then see which max. Index. So this index 135. The reason because I had one intercept. Okay. So the beta value, you see the beta, the slope tells me the relative contribution of your X to predict Y. And this beta, when the position is in 134, actually, because intercepted added, give you the maximize the prediction as a 1. So I can find it. Okay, the beta value tells you the contribution for each independent variable to predictive variable to explain your law. Okay. I think that's enough for this linear regression. Oh, by the way, you see a lot of zeros, okay? Oh, almost 0. Because that single variable explains everything. So you don't have to use other variables to. But if I do 500 to 1000, so I avoid the gene number 134. So this gave me some beta values, right? I hope you know the difference between them. So when I select the gene, not including 134, then the beta value start to show up. If I include that particular variable in there, everything can be explained by that 134 variable, and others will become 0. Yeah, I think that's all for linear regression, and we can take a 5-minute break, and I will come back. We will jump to slide number 51, and we start to discuss the gene expression noise. Okay, question? Yeah. Mm-hmm. Yeah. Yeah. Yeah, you can do that. You can extract from the interface. Let me start an example. Yeah. You click this button here. No, that's one. Yeah. Yeah. Yeah, there's only 23 cells. You don't have to export. You can start directly work on this. Yeah, shut this down. You can close the original. You can start to work on this. This is the only macrophage. Yeah. So you drag this away and close this. That's original. You don't use this. You start to work on the macrophage. You can, yes. Yeah. 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 Oh, you can search for the gene. You want to do the using program here? Okay, I show you. So you know the G, and then you know this gene. You can type name. So you can find G, the name of gene, and then yeah, then you find that give you the ID index. All you can see is number. You put your name of gene here, whether it's a number. If it's yes, it will be one. And then you can also get the index. So there are different ways you can find the index. Yeah. Yeah, so you can try when you have a size, it give you uh, the dimension. So it's a say 1000 by 1. Then once you have the two parameters, it will create this vector. It's all ones. Yeah. You can just type that in. Yeah. Uh-huh. Mm-hmm. Only one G. Okay. Yeah. There's something wrong with it. Okay. So why you want to use one gene to plot it? Oh, violin plot. Okay, I see. Then you should use this, I believe. You can see violin plot. Oh, you don't have violin plot here. Oh, you want to see the for each gene. Oh, I see. Previously, it worked. Really? Yeah. Okay. New version. You can copy this to me, and then I will fix that. Yeah. So you just copy just copy this email to me. Yeah. Yeah. Oh, really? Yeah. 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 Sometimes. And, yeah. So, okay, you want to do the violin plot for different genes. Let's see. Yeah. Gene expression, yeah, but I should gene expression using this. Yeah, you select one gene. That's the way I try. Different groups. Oh, for different groups, I suggest you use dot plot. Type select some genes. Use dot plot, then you have different groups. Yeah. The percentage of expression non-zeros. That's average. So if you have two groups, then you have only two columns, but you have a lot of genes. I think this is better than violin plot. Violin plot, you can only show because they are zeros. That's why the shape not good. Violin plot, the distribution for this is good. The bubble size tells you, for example, 100%. That means all the cells have non-zero. And if a small dot means only maybe 5% have values. So. Uh-huh. Uh-huh. 1, 2. We can 500, that's a lot. But this okay. So if you can give me the 500 gene name, I can build that in here. For example, for example, if you let's see the separate cell types. It doesn't matter. So we can do this predefined cell scores, or you can define your own score here. But anyway, if I have that, I can put a predefined cell score, and then I don't know if I have M1, M2 there. Yeah, but yes, we can build that in there. The one of this. Yeah. Yeah. But anyway, so this is just the name of it. Yeah, you can use this already there. Then you can compute the values for each that the gene use for each category. And yeah, yeah, yeah. Same thing. This is a score computed for every individual cells. So there are cells in there. So that's the marker genes. Yeah. The gene defined as M1. Yeah, yeah, computed. Yeah, yeah. You have two groups to compare the average. Yeah, this will be useful. Yeah. Yes, some of you ask about this final presentation. It's an important part of this evaluation of your learning outcome. So I want you to do is to utilize whatever you learned from this course. Just compare it before you take the course. So you learned something new, and how are you going to apply those methods or algorithms to some proposed single cell data analysis? It can be a data that you already have or you're supposed to have, or you can have your own research question to learn from single cell data, maybe interaction between cell types or other topics you like in the cancer study, in the skin healing, any topic you want to share with us. So the time will be 10 minutes. Then you have 5 minutes for the questions from other students. And your presentation will be evaluated based on the topic you selected, whether significant or the idea is innovation, and your performance to deliver the message, to communicate with your audience. So it's a comprehensive evaluation. We give you a score, and at the end, it will be putting together with your homework, with the presentation, and the final examination combined, give you the final score. Okay. I think it's a time for you after the spring break to start to think of what are you going to talk in the 10 minutes presentation. It can be introduce a new algorithm, can be analysis of existing data, but adding new insights from your analysis. Can be some new ideas. For example, my presentation will be one of the examples. I use single cell data to address a specific question which cannot be addressed before single cell technology become widely adapted. So the question I'm trying to do is to ask whether gene expression noise is there anything to do with the function of the cell, of the gene in that cell. Otherwise, it can be asked, is single cell gene expression variability functionally important? So the gene expression noise is defined as a variability of gene expression for particular given gene in a group of homogeneous population of cells. So in this case, something like this, there's no cell type difference. Okay, all the cells look same. And if you look at this group of cells, if you look at some genes, and they may express very differently from one cell to another. The fold change could be hundreds. Maybe 90% of cells has no expression of this particular gene, but another 10% of cell express this gene extremely high. So the question is whether this kind of genes are important. Okay. So what's your educated guess so far about this question, whether those are important or not? Anyone have you ever think about this? No? No, never. Okay. So this is a paper published in 2015. Yeah, so how can we pronounce his name? Jin-Yong Kim. Jin-Yong Kim. So he's a professor in the University of Pennsylvania. He with his students and proposed this hypothesis or opinion paper. See, variation is function. It's a very strong statement, right? So their hypothesis is the single cell variation is required for this aggregated function. So as a population, you need those variability in order to achieve, as a group, achieve that function. However, you know this single cell technology 10x genomics become commercially available in 2016. So his paper is only, it's opinion paper, it's comments. So let's see what it's called, problems of paragraph or perspective. So it's just a thinking. There's no strong data support. Okay. But with the single cell data, you know, we can start to look at the variability. Okay. So those genes are not so highly variably expressed. For example, this gene. Can't see the name. Let's see. INFS. Anyway, so it looks like there's some noise, but this is not extreme. So this, for me, this gene expression is highly uniformly expressed, basically. You can see it's expressed in all of the cells, basically, and the level is okay, not so drastically different. This is high, but not that high. Not like this, some maybe 1,000 times high. So we need a way to estimate the variability. Okay, see whether the variation for this gene is higher than that, this gene, which one is the highest in terms of variability? Whether some of this have a higher variability compared to, on average, you have a high average, but not necessarily to be the variation. Okay, so we have to consider those in order to compare. So we used existing data set from lymphoblastoid cell line, lung airway, epithelium cell, and the dermal fibroblast. The first cell line data we sequenced ourselves. We have cultured in the Petri dish. And then the other two, we download the data from other people. Okay, they're randomly selected. It was accessible and downloadable, and also they're very different. So that's why we select them. You can select basically any cell type you're interested in to study the variability of gene expression in there. But for this case, we just want to compare by using 3 examples from different cell types. So once we finish this, we will also have a X cell type. I'm not going to tell you what the cell type it is, and when the time comes, you will guess what it is. Okay. So this is a mystery X cell type. So this is something we haven't talked about. Cells are undergone cell cycles, and some cells in the G1 phase, some in the S phase, some G2M phase. In this lymphoblastoid cell line, majority of cells are in the G1 phase. How can we know? There are marker genes which highly expressed in G1, marker genes highly expressed in S. We can easily separate them into different subgroups. And this expression level overall is indicated by the colors. And you can see UMI values is relatively high in S and G2 phase. I believe the reason is the cells, when they have in the S phase and G2 phase, relatively larger compared to G1 phase. Okay, so you have a higher concentration of everything in these two phases, yellower, right? But our definition of a gene expression noise is the cell has to be same cell type and in the same states. Otherwise, it's not a white noise. It's created by either difference in the cell type, subtypes, or cells in a different cycle phase. So that's not fair to use different cell subtypes or cells in the different states to estimate variability. So that's why we will get rid of this. We are not using cells in S phase or G2 phase, only use cells in G1 phase. Fortunately, we get enough cells. We have 6,000 cells, so get rid of 1/3 or 2/3, it doesn't matter. Still have enough cells to estimate variability. Okay, is that clear? So we are using same cell, same states, whatever method we can use to select a group of cells highly homogeneous. So with this data, we remove, filter out other phase, only get this G1 phase cells, remove mitochondrial genes, library size, and we make sure they're very narrow region, have been select the cells in there. And then to do this embedding, okay? Embedding. Then we manually selected a group of cells. I don't have to introduce you again for the embedding, but this picture is for general audience, which has not taken the course at all. So what I try to see here is dimension reduction. This technology to, for example, make a 3D object into 2D, and it doesn't matter how you crash it and reduce the dimension to 2D. They are still same data points, but just look differently, right? So you know, if you do the PCA, use the t-SNE and the FATE, those are different algorithms, you got a different answer. And FATE you can use run 3D as well, of course. But in general, I just feel that's from the same data set. So PCA maybe not separatable, make them separatable. t-SNE tend to give you this a pie shape. Well, FATE tend to give you some branch. So to me, the FATE is highly conservative if you want to select a group of cells homogeneous. Okay, so that's what I want to do. I will embed the cells and only select the cells in this circle. Okay. By the way, they all G1 phase cells. It's already highly homogeneous, but we will just select this. Okay. And those steps are really important to help you to really found homogeneous population of cell in order to estimate the expression noise level. Okay. So so we jump aside a little bit. So see how can we take advantage of these different views of embedding. I told you, I sent you an email. This is a paper published February. And this group create different visualization views. So as in this example, for example, this visualization 1, 2, 3, 4. their method will synthesize all this and generate a new matrix and then do a so-called meta visualization. Okay. So as I said, this function, let's come back to MATLAB here. So I'm loading this example data, but with raw data set. Okay. Remember, you can do the embedding using Disney UMap fit. Now I just provide this meta visualization option. So this is a PubMed, and it's slow because it's going to do Disney first, UMap, fit, and also MDS. And then at the end, we will combine them. So you will get a slightly different picture. So let's select highly variable genes, do 3D. So there are different steps in this. Okay. So the running time is okay for this small data set. Okay, we only have 2,000 cells. So I encourage you to use it and see whether give you the better results. Okay. It will run Disney, and not only one time, but also using different parameters for Disney, using different parameters in UMap. And Disney UMap result will be highly sensitive to the selection of these parameters. Yeah, I will come back to this once it's finished, but I just let you know it's available in there. Okay, so we select cells that's only in this, I call the stem region of this center of fate. Okay, so I believe this is a highly homogeneous population of cells already. Now, if you compare two genes expression profile, I randomly select two genes. One is RPS11, so this is Robertson protein subunit 11. Another is IgH, okay, so immunoglobulin heavy chain. And looking at their expression profile, this bar indicates the level of expression. Those are just randomly arranged cells. You can see 100% of the cells express Robertson protein subunit 11. Okay, but the level is about 100 or okay, the average. Well, in IgHG1, a lot of cells has no expression. It's hard to see, but the expression level in 90% of these cells are 0. No, it's meta visualization doing the UMap, okay?
but those express the cells have values and above 200, a lot of them. Okay, so this create a strong noise for this HG1 compared to Robinson protein. So how can we measure it? We use coefficient of variance, which is standard deviation divided by the mean. So you control for the mean, and then you have CV for this gene 0.43. Well, for this gene it's 6.38. It's much larger, right? By the way, if you do the average expression, they have very similar expression level on average. Okay, so those genes are ranked by their average expression. So the rank of these two genes are very similar, close to each other. Okay, so now we plot these two genes by their mean and the CV in these two places. So they have very similar mean, okay? But one gene have a much higher CV than another. So this will be immunoglobulin. This is the Robinson protein. Then we look at all the other genes and see. Yeah, by the way, this is a slide from a previous PowerPoint. And that tells you if you have, if the gene expression follow this Poisson distribution, you should have in the log scale, you have linear relationship, okay? So negative, so it's negative 0.5. So you expect a line here, okay? This is by theory. By theory, that this Poisson distribution, this line regression in red. So look at all the genes. You have a relationship between CV and mean, looks like that, okay? So what we did in order to estimate variability by control for the mean is we did a regression, okay? Instead of regressed to a linear, simple relationship, we did is nonlinear, okay? So this green curve give you the fitting of this model. So what's on this curve? On these curves, if you have a gene, you know that its expression average, the mean, you will predict its CV. So for this particular gene, for example, its predicted value for the CV is here. But in reality, you observe its CV is much higher than expectation. So you can do this for every gene. So each point is a gene. To look at the residual, okay, so called the residual CV, there's a difference between your observed CV with your expected CV, predicted CV by the regression. So that's place we use regression, okay? We use a nonlinear model. Yeah, so this is a generalized nonlinear model. There are some discussion of what it is, what's the difference, and in the MATLAB documentation, you will find the discussion of this as well. Anyway, so that's what's inside the nonlinear fitting, and you have a model with use a linker function. Okay, so for lymphoblastoid cell line, we that's our real fitting with real data. It's a little bit unfair for highly expressed gene. This regression is not a. It should go across the cloud, but anyway, any genes are way above this curve is their variability is much higher than expected, because your expected variability level CV should be on this regression curve. Well, the genes which below this curve means by given average of that gene expression, its CV is lower than expectation. So all the genes under the curve are those genes non-highly variably expressed, which will, in contrast, the cell genes above this curve are highly variable genes. So this is just a comparison between selected highly variable genes, this 4 and these 2 below. So those 2 are so those are the highly variable genes. Look at their profile, look at the scales of the expression. So once they express, they have a much higher level compared to those uniformly expressed genes. Any questions so far? Clear? This concept will be very important in the single cell data analysis. But what I learned from collaborating with biologists trust or use differential expression analysis too much. That's probably the only way they want to do the analysis. Their final goal is to find differentially expressed genes. But let's see. Okay, once you learn this, you know, DE gene analysis probably is the most last one. I don't want. I want to do. Maybe we can ignore that. We don't have to do DE analysis. It doesn't make sense for the single cell data. Okay. So for instance, in this case, how can you compare this gene with that gene? It looks like the expression level. I mean, let's see. Okay, so in the lymphoblastoid cell line, so we can use this technique to select those genes with the largest residual CV, and those are the genes top 50. Okay, they are not highly expressed gene. They are not in this corner here. So those corner are the gene highly expressed. Let me see, show you. So this part. Okay, their gene expressed highly, right? This is log scale and then highly expressed. So the highly variable genes, most of them are not the most highly expressed. The average actually is not at this end. There are some of the highly expressed, but the majority are in the middle. Then those genes, then we run enrichment analysis and found, okay, those are the enriched term, okay, in different pathway. And you can see, okay, there are cytokine-mediated signaling pathways, immune systems, of course. And overall, so we have impression of this is immune cell. Okay, so this can give you an impression. It's not specific, but in a large ballpark, it's immune cells. So you ought to mix this up with skin cells. We'll see. Okay, so this is from a lung epithelium cell, and this shape. In this case, the fitting is much better. And we will select the genes which far away from the expectation, and then most of the highly variable genes are there. So those are the top 50 highly variable genes selected. There are different genes from a lymphoblast cell line. So different cell type have a different sets of highly variable genes, okay? And if you look at the function of this, that's including cell proliferation, development matrix, extended matrix organization, collagen formation. So those are the cells function that specifically define or associated with lung area epithelium cell, right? That makes sense. If you don't know those cells, have no idea what that cell's function, you know, okay, it's in the surface of something with interaction with the environment. It form the collagen and have some immune system function as well as a defense. Let's see the last one, fibroblasts. And you found highly variable genes by residual CV, and those are the genes, okay? You still have the cell proliferation. I don't know if they're immune-related oxygen content, but this is certainly a very specific keratinization, okay? You don't expect keratinization in the immune cells. You don't expect this in the lung epithelium cell. You definitely don't want to have that. But that's in the fibroblast in the skin. Okay, so summarize those. So different cell type, once you look at their highly variable gene and look at their function, they're highly specific function groups. Okay, you want to have these links. As I told you, this one is very different. Can you see the difference? Can anyone tell me the difference between this picture compared to the 313 we previously see? Yeah, so visually tells you there's no genes on this way above this curve. This almost perfect fit by this regression line. Every gene follow its expectation, so there's no extra level of the variability in there at all. Basically, every gene expressed homogeneously across cells. So what is this cell type? Anyone tells me? Anyone can tell me? No? You haven't, nobody heard about this story? I presented this somewhere already. Say that again. All gene present, there's a lot of genes expressed. Some genes expressed level is high, so they're healthy cells.
Low variability.
Yeah, the variability is really low for all the genes. There's no biological variability in there. Yeah. Okay. So those are so they compare the different shape. So okay, so that's the answer. So this answer tells you when these cells can have the potential to differentiate into any different cell types itself. When the cell at this stage, there are no gene expression noise. All the gene express uniformly across all the cells. Yeah, so just nothing. Okay, we tried, just nothing. No enrichment terms. So this is just a summary, and because there haven't been differentiated into any specific cells, so they are not registered themselves to any particular function. So you don't need any gene or any set of gene to be expressed highly noisily in order to achieve any goals, because they are not ready to be functional to any direction yet. So the other way around is, if you look at the highly variable genes in those differentiated cells, those genes should reflect what those cell types are working on. Okay. So if that is true, we can test this, because stem cell can be derived into different cell types. For example, if you go neural stem cells, this is an intermediate step that you can further differentiate them into different cell types in the neuron. So we are going to look at neural stem cells, a picture of this. So what do you expect? Our expectation is the shape of this will be in the middle of this and the differentiated cell type. Okay. Yeah, so this is a picture we get from neural stem cells. It starts to show some of these highly variable genes start to jump out from this regression curve, right? We can try with other cell types. If you have your own cell types, do it. So this is very specific collagen sites. I believe you know, people study nutrition, they work on this. This is not highly abundant. You found hundreds of cells in one sample. But this is a specific epithelium cell in the bile duct, okay? And that's the highly variable gene and their goal function. So it's highly specific. You don't find this from other cell types of epithelium cell. Do you believe they're give you some idea about what the cell types are working on? I don't know. I have to ask the experts. Okay, they have domain knowledge. See whether collagen sites are working on it. To me, they are working on the since the local environment, this hormones concentration change. I don't know whether it makes sense or not, but very specific cell type. So if you look at the review paper and see what all those epithelium cells doing, and they're regulated by corticosteroids for sure. They have to to be able to sense this change. Of course, there are other factors. Yeah, so the take-home message is so now we can use single cell data to look at the individual cell expression variability. That wasn't possible before single cell data become available. So we should take advantage of this. And most important is very useful because you have a different cell type, have a different set of the highly variable genes. And they are functionally related. That's why it's important. Okay. So when these highly variable genes are important, when you compare the mean across different samples before treatment, after, then you actually, the gene set you get will be completely biased if you don't consider the variability change at all. Yeah, believe me, the DE analysis will drive you to a wrong direction. I have to tell this to all my collaborators from biology labs. I have to write a comment paper to discuss this further. So what you can do is probably we should develop method to identify this gene, change their highly variable states from one condition to another, whether they're increased level of variability or decreased. Okay. So that will involve more statistic problem. How can you address the change of variability and control for the mean or take mean into account as well? So we need a statistics test, which not only DE, but only DV, differential variability, and plus DE plus DV together. Okay. We are working on that. Okay, so that's my presentation. So that's something, it's longer than I expected you to present in 10 minutes, your final, but something, if you have good ideas or you have good view, or you have your applications very thoroughly present, organized. If you don't have data, you can propose, I'm going to use this 3 data sets to achieve some of this test of my hypothesis. That's something like that. Okay. Let's see my visualization. It's finished. Okay, so this is like a very different view from a t-SNE or UMAP alone. Yeah, I don't know in total how long it takes. Maybe 15. Yeah, so that's my new annotation uses meta visualization. Looks like there's the separate cells into these very tight clusters and separate them further. And I don't remember we had duct cell or endocrine cells in there. It looks like they probably gave you some new cell types because of this further concentration of the same type of cells give you a stronger signal. You can compare this with the T-SNE UMAP annotation. But anyway, so these are one options for you to explore and let me know if this works better than others. Okay. Okay, I think that's all. Time is up, so let me know if you have any questions. Otherwise, we'll see you next Tuesday. Yes, keep. Do you need an extension? I can extend it to next Tuesday. Okay. Next Tuesday. Yes. Okay, I'm going to stop my sharing and recording.